Okay, thank you very much. Thank you very much for the invitation, which I wish would have been enabling a personal talk in person, but this is sort of what we have to deal with. So I'm going to talk to you about a phenomenon called epithelial-mesenchymal plasticity, which turns out to have a very large implication for the physics of cancer metastasis. And I'll try to explain to you, first of all, what is this plasticity, how we can understand it, and why it can offer new insight into, of course, what is probably one of the most important aspects of cancer, which is the fact that when cancer metastasizes, it starts growing throughout the body. That's when it turns into a deadly disease. So I'll start from a cartoon that circa 2013 that you can find in many different biology review articles that indicates what the prevalent view was of how cancer metastasis would work. So on the left-hand side here, you see in green a primary tumor made up of epithelial cells. These are cells that form the basis of most tissues in our bodies, lungs, pancreas, colon. And these cells stick to each other via the cadherin bonds that we heard a great deal about in the previous very interesting talk. And as long as the cancer cells are in this mode where they all stick together and they form just a tumor mass, we have a variety of treatments that are effective at combating the disease. There's surgery, radiation, etc. The real problem arises when cells change their structure and begin to leave this primary tumor. And that's indicated here by this sort of line of cells that is progressing outward, eventually reaching the blood system, and of course, progressing through the blood system through various other parts of the body, emerging from the blood system, and eventually starting to grow new tumors elsewhere in the body. So this is the metastatic cascade. It's a series of processes that all need to occur in order for metastasis to take hold in other organs of the body. And what you see in this picture, as indicated by the cartoon, is that in order for the cells to go from this initial primary tumor and to a secondary tumor, they have to sort of undergo this reversible change in their behavior. They have to go from epithelial, sticking to each other and relatively immobile, to quasi-mesenchymal, which means that they can move effectively, they can de-adhere, they can survive in the blood system, they can emerge from the blood system, and then they have to, in this picture at least, undergo some reverse transition back to epithelial so they can begin to grow and colonize the new environment. So when you look at this picture from the point of view of physicists coming into this field, there are a number of questions that you can ask yourself, which try to make sense of how that picture is supposed to work. So first of all, we can ask, what is the physics underlying the type of cell motility that you actually see in this problem? So we see sort of motility, which is meant to occur at the single cell level and involves going through matrix and eventually emerging in another part of the body. So we'll talk a little bit about cell motility to get started and different models of cell motility from the physics point of view. And underlying the cell motility in this problem is that the cells need to dynamically change their state from one type of cell to another, epithelial to mesenchymal. That state is typically called a phenotype, as distinct from a genotype. These cells are not changing their genome, at least they do not need to change their genome. They actually just change their properties at a fixed genetic background. So how does this occur? What are the signals that give rise to this? What is the dynamics of that transition? And then, of course, fundamentally after all the physics is said and done, of course, one of the issues that motivates a lot of the work in this area is if a better understanding of these issues and better quantitative understanding, a better conceptual understanding can help us better understand and treat cancer. So that's going to be the sort of a structure of the talk. We're going to talk about cell motility. We're going to talk about phenotypic transitions, and then we're going to talk about how this might be relevant in the actual cancer project. So let's start with cell motility. Cell motility is now, actually, you can think of it as a new branch of condensed matter physics, the branch of active media. It's the branch where individual objects, for example, is indicated in this beautiful picture of bird flocks, individual objects that can move in a non-equilibrium manner because they have their own energy sources and they can interact with each other. And the issue is trying to understand the forces and energy sources that give rise to motility and how, under certain circumstances, that motility can be collective, that individual objects can affect each other in some non-trivial way. So this is what I call the world of active media. And we can start by looking at single cells. So this is what a single cell moving might look like. This is a cell which is moving with sort of chemical information where there's a chemical gradient pointing from the left to the right. And this is a cell that is specialized in detecting chemical gradients and moving. And just for sort of the physics of motility, what you see on the right is actin protrusion labeled in red, and what you see on the left is myosin-based contraction, which is labeled in green, and the cell then can sort of morphologically move through, in this case on the surface, but in general move through different materials and progress from one point to the other. And this is the type of cell motility that was indicated in that cartoon, an individual cell moving, maybe guided by some gradients of nutrition in the cancer case, and making its way to the blood system. But this is not the only type of cell motility. We can also very often see, as I mentioned already, collective cell motility. This is now cells, but it's similar in some general sense to the bird flocking problem, where we have many cells. These are actually dictyostelium amoeba in a rotating aggregate. And the basic physics at the single cell level is the same. The cells still have the same propulsion generated by ATP. They're still governed by adhesive interactions with the substrate. But the difference now is that in addition to those individual cell interactions, they have cell-cell interactions. Those cell-cell interactions determine collectively which way the cells are going to move. And this idea that you can have these non-equilibrium quantities interacting in large statistical ensembles, giving rise to a variety of patterns and structures, gave rise to the entire field now of active media. And for those of you who want to see what the field came from, there's sort of two seminal papers that sort of defined this field in 1995 and 1998. You can go and look up this thing. And this particular picture is from a paper we wrote already in that sort of early era in PRL in sort of the year 1999. Now, of course, we're in the business of theoretical physics, and as theoretical physics is in the business of making relatively simple models that allow us to analyze what is going on, decide what are the important mechanisms. So over the last few years, we've created a variety of different models of looking at how cells move collectively, also depending on the exact experimental realization, cells also divide. So you have cells that are moving, that are dividing, that are interacting with each other. And a key notion, which I've already sort of mentioned in the previous slide, a key notion in these models, aside from the forces that cells exert through their adhesive context with the substrate that you can model based on many experimental measurements of those forces, a key notion is how a cell is going to polarize. So a cell is going to pick a front and back. You see that over here in this picture. The red, again, is regions where actin is polymerizing and pushing the front of the cell forward. The green is where myosin-based contraction is pulling the rear in as the cell moves. And one of the major issues in collective motility has to do with how is this polarization of an individual cell affected by the polarization of the neighboring cells. And just to give you an indication of one of the biological effects that are important, there's a notion called contact inhibition of locomotion, which says that if you are essentially running, yes? Question?
No, they are not conferences.
Well, I can't
Seminar,
quite.
that is also not a workshop. These are outreach activities.
Well, I can't hear what's going on. Okay.
May I request everyone else to mute their microphones, please? Please mute your microphones. Thank you. Please continue.
Okay, thank you. Yes, so one of the major forms of interaction, just to give you an example of how cells affect each other, is called contact inhibition of locomotion, CIL, and that's a signaling-based network that you see in this cartoon picture over here from the work of Andrei Levchenko, that says that when cells try to move into each other and collide, essentially signals are exchanged between the cells that convince one of the cells to turn around and go the other way. So contact inhibition of locomotion says essentially that cells, when their motion conflicts with their geometry, when they try to move into each other, there will be a dynamic rearrangement to repolarize the cell, and that effective interaction will give rise to these ordered collective patterns of the type you saw in that rotating aggregate. So we set out to create a model which would be able to encompass this. So just to give you some ideas of what this model can predict, if you look at just spreading tissue, so here we have just an initial set of cells, and these cells are going to move outward. They move outward because if they try to move inward, they collide with each other, and then they get reoriented. If they move outward, there's obviously no boundary, no impediment to their motion. So the cells dynamically switch their polarization on average to create some tissue that's expanding outward. Of course, as you expand outward, you have to have cell division. Otherwise, you create gaps in the middle. So this is what a simple simulation of that would look like. And there are interesting things about the simulation. One thing you can see immediately from the picture is that there's a density gradient. So the density is higher in the center than on the outside. That's because the outside cells are moving quickly and moving outward, whereas the individual cells are since jammed in. Something which you can't see directly, but which you of course can measure in the simulation and also measure experimentally, is the mechanical state of these cells, and that's indicated by the top graph on the right. And what you see on the right-hand side is that the cells in the interior are under tensile stress. They're pulling each other, and the tensile stress builds up towards the interior. So the interior is under the maximum tensile stress, and it decays as you go into the boundary. Now, the reason we focused on this simulation is because the nice experiment started already about a decade ago in the group of Dave Waits, and now carried on by Xavier Trepat in Barcelona, showing that you could look at these tissues, you can make measurements of the forces, and you can actually derive, this is from their experimental paper, that there is an increase in tensile stress as you go into the interior. And in fact, the tensile stress is correlated with the cell density. The density goes up at the same point that the tensile stress goes up. This is very non-intuitive. And in fact, the reason I got into this subject is by looking at that graph and saying, how can it be that you're increasing the cell density and yet the cells are under more tension? And the answer is the interplay of contact inhibition of locomotion and the motion of individual cells will actually give you this effect. So I mean, this is a long subject matter with many, many people. So other experiments we'll just skip that. But I want to return to what this implies for the problem which I'm talking about today, which is the problem of tumors. So what we've seen is that cell motility comes in a variety of types. There's individual cells. There's also collective cell motility. And what the biologists have argued is that cells at the margin of the tumor become motile, and that involves, of course, a reduction in cell adhesion, remodeling of their internal cytoskeleton to allow them to move away from the primary tumor. So if I take the model that we have and just apply it to that geometry, then of course, it depends on the relative strength of the motile forces, the forces exerted by the cells on their environment that allow them to move versus the adhesion forces. And in general, the picture that the biologists have are indicated by the leftmost simulation, where you have this edge of the tumor. This is periodic, so there are two edges. This is sometimes called a wound healing geometry. But what you see here is that individual cells leave and move independently. It's sort of like a gas of cells boiling off the edge of the tissue. But that's not the only possibility. As I already indicated, if you actually have stronger adhesion, then cells can still move, but they can move collectively, and moving collectively would look more like an interface motion problem. So now the cells would, of course, still move, but now instead of moving as individuals, instead of boiling off, they would actually move as sort of a liquid spreading out at the edge of a tissue. And this depends on the properties of the cells, depends on how strong adhesion is versus how strong the motile forces are. You can also get other structures. This again is the edge of a tissue. This is now a collective structure, but not just a planar interface, something which you can think of as a finger or as a sprout moving outward. And this type of structure is created by actually having specialized cells at the edge called leader cells, which guide the motion outward. It's again a style of collective motility, which is a little bit different than the previous style. And again, this depends on the details of the biology, the strength of various interactions created by these changes in the cell at the edge of the tumor. So the lessons so far from this whole field, which I've very large field now of active matter and active matter using cells as a primary example. The lessons are that cells are self-propelled objects that can organize their motion cooperatively under a variety of cases. That means that one can expect that some cells will move individually, as in that original cartoon, but others will move collectively, either just by expanding the whole tissue or under other circumstances by sending out sort of streams of particles away from the primary tumor. And this is controlled by biophysical parameters such as cell-cell adhesion versus the strength of propulsion forces. So now we have to go back and ask, okay, let's go back and look at the cells at the edge of the tumor that are supposed to be changing their phenotype from one state to the other. And how can we describe that? And how can we understand under what circumstances we're going to expect one type of motility versus another type of motility being relevant for metastasis? Now, before doing that, I want to focus on one experiment that I really liked, because I think this experiment really has a very important set of lessons for thinking about what really causes this phenotypic transition, why cells are changing things. So this is an experiment done in melanoma. And what you see here, again, as a cartoon, is you have this tumor, which is growing. That's what these green cells are, moving from left to right. You see this tumor growing. And up to panel number 4, the tumor is growing in place. None of these cells are detaching from this primary melanoma tumor. And then what happens is the cell reaches a new region of the tissue. It goes upwards, and indicated again in the cartoon by red is a new chemical environment. So the details of this chemical environment are very interesting, but beyond the scope of what I want to talk about here. It's some signaling molecule that is prevalent in this environment. And this signaling molecule interacts with the cells that reach this regime. And that signal is what causes the cell to change their phenotype. So the lesson is a couple. First part of the lesson is that genetic changes, mutational events, can be the cause of changes in the cells, but need not be the cause. Here, nothing is happening to the cells per se that is genetic. They're not undergoing new mutations. There are no new clones. This transition would not be detected by spending lots of money doing cancer genome sequencing. This is purely a transition that's induced by a signaling event caused by changing the local microenvironment in the neighborhood of the primary tumor. And in fact, that's what we think is most typically what goes on, because years and years of effort that try to identify specific genetic changes that must occur that would give rise to this type of invasion and spreading and metastasis have not really been successful. It was thought initially that there would be some very specific genes that would be enabling for metastasis. If we could find those genes, we could find chemical inhibitors of those proteins, and that would be a great way to treat cancer. And the problem is that's probably not the right way to think about it. The right way to think about it is you're switching a cell to a behavior which is natural for the cell, becoming motile from immotile, and it's not occurring by some very easily targeted individual mutational event. The other feature that's interesting from this is that the change here is somewhat irreversible. The cells reach this region, touch this region, change, then they leave. And when they leave, they actually have not reverted back to their original phenotype. So whatever this signaling changes, it has the capability of creating irreversible changes in the cells. And so then the cells leave this region and they go on and metastasize in other parts of the body. So the issue for us when we thought about this problem based on data like this and our concept is that we need to think about biological networks. We need to think about how biological systems enable different states under sort of fixed interactions. So in this field, there's a variety of pictures that sort of indicate the types of networks that one is interested in. This is a picture from Rika Albert in 2015. And this is a very interesting picture because I think it hits the sweet spot where every physicist looking at this picture says, wow, this is so confusing, we'll never figure out anything. And every biologist says, amazingly, how much of the detail you've left out of the problem. How can you even learn anything by looking at such a reduced representation? So this is exactly in the middle of that spot, which I think is why it's a nice picture. And I want to indicate two features of this. This is actually the circuit that is supposed to give rise to that type of transition that we just saw in that previous picture, where cells receive some external signal and they change their state in response to that external signal. And the transition is called this epithelial-mesenchymal transformation. This is a simplified EMT circuit diagram. So I want to just call your attention to two parts of this diagram. Just one part up here, this is just the input, one of the inputs to the system. And what you see here are all these signaling molecules, which go by the name of notch delta system, various growth factor receptors. This is the Wnt signaling system, all the signaling systems that give input to the cell. And this is just one particular aspect of it relevant for that experiment, a notch delta system. But then down here is sort of where all these inputs converge onto a set of interacting factors, snail, twist, zeb, which we'll talk about in great detail on the next slide, where the decision is made. So you can think of the circuit as some type of logical diagram where inputs come in, there's some type of pre-processing of those signals and mixing of information from different channels that we probably don't understand at all. And then eventually the signals emerge to a sort of a computational module, a sort of decision-making module down here. And then of course there's outputs such as the strength of the adhesion, for example, given by the expression level of a cadherin bond, and also other factors that affect the actual behavior of the cell. So if one focuses in on that core decision module and looks at it in numbers of different cells, we identified a sort of, in some sense, the sort of guts of this core module. And the guts of this core module was a series of coupled switches, so two coupled switches, giving rise to, on the one side of the switch, to mesenchymal-like behavior, on the other side of the switch to epithelial-like behavior. Now, again, there's very important issues of this circuit. First of all, snail and zeb. This side of the switch, when this side is high, you're in the mesenchymal cell state. So snail and zeb are typically called EMT transcription factors. These are transcription factors, which means that they bind DNA and they affect gene expression of all sorts of downstream effectors. And as I already said, some of those downstream effectors are, they suppress adhesion, they change the polarity of the cells to allow for motility, they change the cytoskeleton, all sorts of things that are part of this transition from one cell type to another cell type. And snail and zeb are snail is sort of the input part, and zeb is sort of, in some sense, the final arbiter of the decision here in this circuit. And when these are high, you get mesenchymal snails. Now, of course, biology is always more complicated than any simple model like this. There are multiple types of snail, there are multiple types of zeb, there are other intermediates. And all that is fine. We'll talk a little bit about extending to more realistic circuits in a moment, but let's sort of try to understand what the core behavior is. On the left-hand side, the thing which is the epithelial side, in other words, the side which, if this side is high, and of course high mean, these things are mutually repressing each other as switches. So if this side is high, we remain in the epithelial state. And what was interesting to us when we first looked at this problem is that this side are not transcription factors. This side are small RNA molecules. This is microRNA 34, actually a family called microRNA 34. This is microRNA 200. This suppresses these transcription factors, but suppresses the transcription factors not by interfering with their transcription, but actually by interfering with their translation. So it turns out that there's a whole new biology since which I call biology since I went to high school and learned biology, which has to do with the fact that regulatory networks in mammalian cells are not just composed of protein transcription factors, which is what people, for example, studying bacterial systems such as E. coli, or even lower eukaryotic systems such as yeast have focused on, but also sort of equal weight are all sorts of RNA-based regulatory mechanisms. These things are called microRNAs. The way they work is they eventually create a 21 nucleotide small piece of RNA. It goes and binds to messenger RNA. That's what this sort of picture is here. These things bind over here, and they interfere with the translation of the messenger RNA into protein. Now, it's a very good question to ask why this method of regulation is somehow better or worse or different than regulation at the transcriptional level. Again, that's an interesting question, but from a more practical perspective, that is what happens in this circuit. So one needs to understand exactly what the dynamics are. We started looking at this a long time ago, but the way we do this, I don't want to bore you with the details of all the equations, but we do this using, again, a sort of quasi-equilibrium approximation, which is actually a generalization of what's called in the transcriptional regulatory field the Shea-Akers formalism. Essentially, here is a picture of our messenger RNA. And these are these small microRNAs that come along and bind to it. And in general, there can be multiple binding sites along a single messenger RNA. And each of those binding site configurations has a certain equilibrium probability, assuming fast on and off rates. And each of those configurations gives rise to a certain amount of additional degradation of the messenger RNA and also additional inhibition of the translation. And you can look at various experiments that genetically modify these binding sites to change the number of binders to get some insight into what these coefficients are. And you can create a sort of dynamical systems model where you have equations for the microRNA concentration, which of course affects the distribution of these binding sites, the messenger RNA concentration, and of course the protein production for a given target. And these can then be folded into the overall circuit equation to give rise to now a circuit which contains regulation both at the transcriptional level and at the translational level. Okay, so now when we did this, what we discovered, and this has to be stated in some careful way, what we discovered is that these circuits, in terms of typical parameters, so not every parameter, not always, but in terms of typical parameters that one would get from looking at experimental literature and making sort of reasonable assumptions, was that this circuit could clearly support more than just the two states that people had sort of been tacitly assuming for many, many years when they looked at this type of problem. So as I said, the cartoon in the cancer biology literature, and that cartoon is sort of a stand-in for 1,000 papers, had tacitly assumed that the choices for the cell would be either purely epithelial, which would be down at this end of the phase diagram, or purely mesenchymal, that's up at that end of the phase diagram. And that therefore, when cells underwent this EMT, they would automatically find themselves in this individual cell motility frame, where mesenchymal cells, typically like immune cells or fibroblasts, are ones that are loners. They move by themselves. But what we discovered was that under a variety of, of reasonable conditions, cells could exist in what we called a hybrid state. So we labeled the state E/M. So it's a state which is a partially empty state or a hybrid state. It has properties which are a mixture of epithelial properties and mesenchymal properties. So the mixture of properties means that it still retains a significant amount of cadherin-based adhesions. So cells still can stick to each other quite strongly. But on the other hand, the cells now become rather motile. Their cytoskeletal organization has changed. They no longer have up-down polarity. They no longer have various other features of epithelial cells. And so now they can move much more effectively. And this type of phase diagram basically says, well, if you're at some low level of excitation, which is just indicated by the x-axis here, then of course you're going to see only epithelial cells. If you're some very high level of excitation, you'll see purely mesenchymal ones. But in the middle, there are a variety of possibilities. You might be in the bistable regime where you see mesenchymal ones and hybrid ones. You might be in a tristable regime where you see all three phenotypes. And of course, you can detect these things by looking at various markers that are indicative of being in these states. So when we first saw this, the first thing to say is, well, does this make any sense at all? Is this a possibility that you can see in the lab? So we collaborated with a Hanash group at MD Anderson Cancer Center. They had a series of 48 different cell lines, lung cancer cell lines that they were investigating. And they were doing basically proteomic scans of these cell lines. And what they discovered in their proteomic scans were that, some cell lines looked like they were very epithelial, that all the proteins they saw looked like epithelial proteins. So for example, this tall lines here, CDH1 is E-cadherin, the standard cadherin for epithelial cells. They saw other cells over on the right-hand side where they saw very little E-cadherin, but they saw large amounts of vimentin. Vimentin is an intermediate filament relevant for the cytoskeletal organization of mesenchymal cells. And then they saw a whole bunch of things in the middle that they didn't quite know how to interpret, where it looked like you had these cell lines that were exhibiting both mesenchymal proteins and epithelial proteins. And then, so we pointed out, of course, that this was very interesting, but we had to first of all distinguish between two possibilities. These are bulk experiments where you just look at a large number of cells and you look at their proteomics, and seeing these mixed behaviors here can either mean that you have mixed populations where some percentage of your cells are epithelial versus some other cells are mesenchymal, or it can mean that you actually have individual cells that exhibit both proteins and you can't tell the difference in a bulk measurement. So we worked with them to look at various of these cell lines, and we picked, here's just one example, where we look now at immunofluorescence staining at the individual cell level, where we actually looked at, again, fluorescent markers for E-cadherin and vimentin. What you see on the bottom left over here is an example just as a control, from the purely mesenchymal end, where you see only green. So that's one of these cell lines out here. Here you see both a mixture of green and red. In fact, a very large percentage, 80–90% of the cells exhibit both green and red markers. So what you see is, you see at the single cell level, you can have cells that are in this type of hybrid state. And we, of course, were ready with a prediction as to what would one of the major biophysical predictions that you would see if you're in this hybrid state, because if you remember my motility simulations, we know what we expect for a purely mesenchymal state. Here is this wound healing geometry where you do this experimentally, and you see these individual cells boiling off the cut tissue. On the other hand, if you do the same experiment for these hybrid cells that retain significant adhesion but nonetheless become motile, what you see is you see the motion of a front here from one closing the edge effectively, but closing it not as a gas, but closing it via this moving, relatively coherent interface. So not only can we identify these hybrid cells, but we can immediately show that the simplest properties that one would predict for these cells were in fact indicated by these cell lines. Now, we also developed a number of other experimental tests for this idea of hybrid cell types. One test, of course, is based on the fact that, as I already said, our model is ultra simplified. There are lots of other things that couple to it. So we identified a number of other proteins that would couple to our circuit. One very nice one is called GRHL2, which turns out to be very important in the development of epithelial cells. So GRHL2 is something which suppresses ZEB. And so therefore it tends to favor the epithelial state. But if you knock out GRHL2, what happens is cells that otherwise would be hybrid are now pushed all the way to their mesenchymal limit. And so if you take that cell line that was moving as this sort of collective motility hybrid cell state, if you knock out GRHL2 or some other ones, oval, then in fact you can make the cells transition to where they're now moving purely mesenchymally. So we could predict a series of factors, which we call the phenotypic stability factors, which if you changed the level of those factors, you could change the phase diagram, push the cells into a different state, and that would serve as a validation of our model that predicted which structures arising from the genetic interactions would be corresponding to these hybrid cell types. Now, now I'm going to turn to what the biological consequences of all that are in a moment. But before doing that, I want to point out that it's always when you do these networks, you always have to ask yourself, well, I'm looking at some ultra reduced representation, only a set of simple factors which seem to be important, but how many of the results are going to be maintained when I look at increasingly sophisticated circuits? So here is an example of a sort of next level up circuit, which is now more realistic. So now you see ZEB has become ZEB1 and ZEB2. Whoops. Snail has become Snail1 and Snail2, and also twist. Now we have microRNAs occur in not just one type, but these different families. We have some new players in the system. We have this one I call the oval, GRHL2. We have other players in the system. And this is sort of I said, the next level up of complexity. And now we needed the methodology to sort of analyze these circuits. So we developed a sort of random perturbation strategy where we sort of looked in general at what are the states that are allowed in some network as complicated as this. And on the right-hand side, what you see is a diagram of sort of the typical behavior that this circuit can actually support. And again, we could show that it supports epithelial states. Those are the ones on the top picture here, where some number of genes are blue and some number of genes are red. It could support mesenchymal states, which is sort of the flip, where all the ones that were blue previously are now red and vice versa. And then it also could support these hybrid cell types in the middle, where one got these mixed expression patterns. And the other thing about these models is it then gave us the ability to create bioinformatic tools that now allow us to look at actual data from cells and determine, based on expression patterns, what type of cells states we expect. And the last part of that I wanted to mention is a paper we continue to work on this in our latest work in 2020, which shows that these networks have interesting organizational structures. That was the 26-node EMT network, was the picture I just showed you. And the interesting thing about this network is that if one takes the network and randomizes it, one finds that there's a huge number of different possible states, in the neighborhood of thousands or whatever, with no particular structure. But if one takes the actual structure of the network as measured in many biology experiments, they're actually very specialized states down here. This is some measure in what we call the frustration of the different states. You see this tail down at the bottom. These are low frustration states, which are the specific phenotypes that are allowed by the specific connections of the network, and these turn out to be the most dynamically important. And so there are interesting structures in these larger networks that in some sense support the idea that one can learn about what's going on by reducing the system to some simplified network, because of this structure that many of the additional interactions of the system actually are relatively consistent or relatively non-frustrating compared to what would be the case of a random network with the same number of links and nodes with the same topology. So those of you interested in that, I suggest you look at this last paper in PRL that we published on this issue in a number of different networks. So if I go back to the biology, again, I showed you that this type of state could exist in vitro cell, just taking experiments in the lab and letting them move on surfaces. But of course, more interesting is, do these type of cells exist in real cancers or real patient tumors? So an experiment done in 2008, 2017, this was for head and neck cancer. They took detailed expression patterns from human tumors, and this is their classification based on their particular, genetic markers that they thought were indicative of these different types. And inside the tumor, in the main bulk of the primary tumor, there was no EMT, so that means these cells are epithelial-like. Then the cells at the edge of the tumor, they actually showed were mostly in this partial EMT phenotype, and then full EMT, the ones that were really mesenchymal. In fact, there were very few of them, and in fact, most of them weren't even cancer cells. There were other cells like fibroblasts that, of course, were, initially purely mesenchymal. So they argued, based on a human tumor, that in fact, the picture of the edge of the tumor changing its phenotype was valid, but that it didn't change to fully mesenchymal. Actually, in this experimental system, it changes mostly to this partial mesenchymal state, partial EMT state. And then another version of the same type of result from pathology paper, where they went and took out tumors and looked at the cells in detail, and they looked at clusters of cells leaving the primary tumor. And the important thing in this experiment is twofold. First of all, what they saw leaving the primary tumor were typically not single cells, were clusters of cells, which is indicative of some type of collective organization of these cells that are moving. They are sticking to each other. So these clusters were relatively small, but nonetheless, they showed clear evidence of adhesion. And in fact, the people who wrote, the individuals who wrote this paper actually noticed that cells in these clusters express ZEB1, so they look partially mesenchymal, but they also still have E-cadherin, so they look partially epithelial. And they hypothesized that this was some type of partial EMT phenotype, and this was done independently a year after we sort of introduced this notion in our circuit model, but it's completely consistent with not only what we expect to see, but also the specific prediction of what the molecular signature would be for these hybrid cells. And finally, in a paper in 2013, this was a tumor. This is now clusters of cells seen inside the blood system called circulating tumor cells, where one goes to the blood of a patient. And of course, if the cells are making it from the primary tumor to the secondary metastatic sites by going through the blood, it means that if you look in the blood, you should see those cells. So they did that in detail, and they were actually able to show that there were clusters of cells in the blood. Those clusters of cells did consist of cells that they called E=M, which meant that had sort of balanced epithelial and mesenchymal properties, and that in fact, the more aggressive a tumor was, the more likely it was to metastasize, the higher the percentage was going to be of these sort of clusters in the blood. So this point of the story, of now just the circuits and dynamical systems that are giving rise to these phenotypic transitions, what we've seen so far is that cells do undergo motility transformations at the edge of the tumor. You can see that experimentally in a number of different systems. You can see that in terms of the properties of the single cells and the signals that they're receiving in the human microenvironment. This, depending on the circumstance, this might create individual moving cells, full EMT, or collectively moving cells, partial EMT, and evidence is sort of increasing that partial EMT is not a rare occurrence. It's actually common, maybe even more common than full EMT, and leads to formation of clusters of these metastasizing cells rather than individual cells. Okay, so that brings us to the last part of the talk, which is, so what? why do we care? Why do we care if cells leave individually versus leave collectively? Is there something special about these clusters? Is there something special about this phenotype that enables us to better understand and maybe eventually even treat cancer? So let's turn to the sort of third world. We had so far the world of active media. We had the world of nonlinear dynamics and its relationship to genetic circuits. Let's now talk a little bit about actual cancer research. And so let's return to this 2013 paper and its follow-up in 2014 of these clusters that were seen in the blood system, which, as I already argued, are indicative of an EM, a hybrid phenotype, and indicative of the fact that that phenotype allows for the formation of clusters. So in a follow-up experiment, this group at Harvard and Mass General Hospital and a separate group at Johns Hopkins asked the question of how did the ability of these clusters to form metastatic lesions compared to the ability of single cells. So in their blood samples of patients and breast cancer patients, they saw both individual cells and clusters. And so they could then go via some very clever labeling of cells, which I won't go into because I don't have time. They were able to calculate in a mouse version of this experiment. So they redid the experiment in a mouse. They were able to calculate effectively how likely was it that a metastatic growth emerged from a cluster versus emerged from a single cell. And so here is the ratio. It says that there's a 50 times, or in some other version of experiment, 25 times more likely for a metastatic lesion to emerge from a cluster compared to emerging from a single cell. Now, one has to normalize this a little bit because clusters have more than one cell. Typically, their clusters had, on average, maybe 5 cells per cluster, maybe a little bit more. So a factor of 50, even if I normalize it on a per cell basis, is still 10 times more likely for an individual cell to be able to start growing again, whether it's part of a cluster of other cells or whether it's just goes off by itself. So this transforms the problem of the formation of cluster into something which is much more relevant, because now we see that clusters can really be an essential component, can be a major pathway for enabling an individual cell to start growing in a new site. Now, this was then shown in more detail in a different experiment published in Nature in 2018, which is, again, a mouse model. So this stands for genetically engineered mouse model of, this is now some type of skin cancer, squamous cell carcinoma. And what they did is they grew tumors in a mouse. They took out the cells. They labeled them in terms of their EMT properties. So again, with their own exact way to do this. But here they took cells that were very epithelial-like. They took cells that were very mesenchymal-like and arranged them along some axis and classified all their cells. And then they re-injected these cells into the mouse and asked which of these re-injections were going to give rise to more metastases. So which cells are more able to actually generate new metastatic lesions? And what they stated was that the most metastatic cells were ones that were hybrid. They were not purely epithelial, but they also were not purely mesenchymal. They were ones that somehow were in the intermediate. And they stated, well, it was particularly exciting to observe that in contrast to what one would expect, the tumor cells in the early stages of EMT with intermediate epithelial and mesenchymal phenotypes, rather than the tumor cells that underwent complete EMT, are the most metastatic populations. So this paper is fantastic, and there's actually follow-up work by the same group that sort of supports this. The only statement I disagree with here is the statement that says, in contrast to what one would expect, because by the time of this study in 2018, we had actually published several papers exactly predicting this effect. And so they even cited our papers, but they didn't quite understand what we were saying, which is part of the continuing struggle in this field is communicating ideas to people who are not sort of, as has much background in sort of this type of modeling systems. But in 2015, we asked the question of, how did this EMT transition relate to what's called tumor initiation capability, the ability of cells to go to a new part of the environment and start growing. Now, if you think about cells going to a new part of your body and starting to grow, it's almost a miracle that they can do that, because cells become very specialized. Cells that are in your liver are specialized for being in your liver. Cells that are in your breast are specialized for being in your breast. All of a sudden, you take this cell and you move it to, let's say, your lung or your bone, and the chemical environment is different, the mechanical environment is different, everything is different, and yet the cells can somehow regroup and be able to start growing again. And so you ask, how can they do that? Well, the answer is that cells have to be plastic. They have to be adaptable. They have to be able to turn on parts of their machinery that allow them to change all sorts of their physiology, their metabolism, their way they stick to their neighbors, their reaction to mechanical forces, in order to adapt themselves to this new environment. Well, that ability to be plastic and adaptable is, in the context of cancer, is sort of the tumor initiation capability. In the context of other cells, is sometimes called the stemness of the cell. How fixed in their ways are the cells versus how malleable they are to respond to different signals and to change the way they behave. And of course, we know that embryonic stem cells are the most malleable. Depending on the signals they receive, they can go a whole bunch of different ways in forming viable cell types. So we coupled using our general approach, we coupled our EMT circuit to circuits that control the stemness of the cells. And here is, for example, OCT4. This is just a simple indicative circuit model. OCT4 is one of the standard factors that give rise to stem cell-like behavior. And we came up with a picture which looks, this was not our picture, but it very nicely summarized the results of our study, was that cells in this sort of intermediate stage, the ones that were partial EMT, were in fact more likely to exhibit this plasticity, this stem-like behavior, or initiation capability, than cells at either end. And the logic is not that hard to understand, that cells that are very epithelial are highly specialized, they're differentiated. Cells that are very mesenchymal are highly specialized. As cells move towards the middle of this regime, this window of stemness, they become less differentiated, they become more malleable. You can see this in any one of a number of ways, including, for example, with the epigenetic structure of the cell, something I'm not going to talk about. And therefore, these are the cells that are able to go into a new environment and eventually adapt themselves and start growing. So we could predict that the cells that were in these partial states, not only would they change their motility characteristics, but would actually change their ability to actually form new metastatic growths. So we would predict what was seen in those original cluster experiments, that the cells in the cluster were the ones that had these special properties that enabled them to actually form new metastases. And again, now, sort of things are catching up to this idea. I already showed that paper in Nature in 2016 already. In some other experiments from Bob Weinberg's lab, they again showed that cells that only had undergone partial EMT is best positioned to acquire stem cell properties. And then in a follow-up paper in 2019, they actually showed that this tumorigenicity, this ability to form new tumors, is actually dependent on having cells in this hybrid state. And so the details are interesting and we can talk about it, but that's the point, is that this picture now has sort of changed what people think of as the importance of EMT. And of course, it also changes what one should do to counteract this, because just having treatments that affect cells that become purely mesenchymal is not useful, because treatments actually have to target these plastic cells in order to prevent metastasis. And now there's a real problem, which is very hard to solve, because not only are these cells adaptive to new microenvironments, but they're also adaptive to treatments. If you give this cells a drug, cells that are highly differentiated can't cope and die. Cells that are in this naturally plastic adaptable state are very often able to find solutions to the presence of the drug and actually able to survive. And in fact, hybrid EMT cells are more resistant to chemotherapy, and actually now in most recent data is more resistant to attack by the immune system. So it makes it a very hard problem, but at least it tells us what the right target is. So the take-home message of the last part is, well, we've predicted these new types of cell phenotypes. These move collectively, leading to clusters. And most importantly, these clusters seem to be a major contributor to the ability to grow new secondary tumors, and hence should be a priority for proposed treatments. So one of the sort of underlying lessons here is that this is a very hard problem. It's not like one can just sit down and solve the problem of cancer metastases. It's not like we've solved it. It's just that we've made some what we feel are interesting conceptual advances that are changing the way the community thinks about the problem and how they go about trying to deal with it. And this has required what I think of as sort of a many worlds interpretation of how to make progress. We needed to combine ideas from soft matter physics, cell motility, active matter, which is a very lively field of endeavor. Many interesting papers here on all sorts of different aspects of this. There's a totally separate field, information processing via biological networks, how genetic networks take signals from the outside and make cell fate decisions based on those networks, and how all the physiological aspects of the cells are sort of coupled together to enable those new phenotypes to thrive. And then, of course, we need to take information from what cancer biologists are doing, who are, of course, the ones who are making measurements in both in vitro and in vivo systems to try to see to what extent the ideas that one can create might make sense in reality. So this talk is focused on one aspect of that problem, the role of hybrid phenotypes in getting exactly the Goldilocks combination of properties to effectively metastasize. This may be relatively rare, but it doesn't matter because there are many cells that are shed from the primary tumor, and it only takes one or a few cells to actually start a metastatic growth. So rare cells are the important ones to deal with if they are the ones that are responsible for the primary number of tumors. And have we made progress? Well, at the scientific level, yes? Yes? Oh, at the science, so have we made progress? At the scientific level, I would say the answer is yes. And at the clinical level, well, I think the answer is still, it's still under investigation. This can lead to some, whether these new ways of thinking can lead to some new treatments and new strategies for actually dealing with cancer metastases, which, as I said right at the outset, is really the cancer metastases, and the fact that metastases are not really treatable by most of our existing therapies really is the medical problem of cancer research in many ways. So I'm done. Thank you for your attention. I want to point out the two groups of people that are essential for this work, and this is only a subset. And the two groups are very different. The top group of people are a whole bunch of people who run cancer biology labs in a variety of places at MD Anderson Cancer Center, at Johns Hopkins, at Duke. And these people have been incredibly forthcoming in sharing their ideas and sharing their positive suggestions, sharing their criticisms when we've gone and talked to them about various experimental pieces of data, about various new experiments that could be done to test some of these ideas. And this work is not possible to do unless you have the sort of active collaboration with leading cancer biology labs in a number of different areas. And I've talked about some of the work related to this, but these are sort of some of the major players that have sort of helped us with that. And of course, the other side of the coin is that I get to talk about this, but of course, other people get to do the heavy lifting. And here's just again a subset of people from a group I've worked in who have had major impacts on some of the work I talk about. Mohit is the person who will talk next when he takes questions, who's of course now in Bangalore. Mingyang Liu, original postdoc, is also on the faculty of Northeastern with me, and Shuafei Li has also moved on to a faculty position back in China, and a couple of people who are still at the postdoc level, and Shubham Tripathi is a graduate student who actually is now working on these, among other things, on these larger network considerations, and was the primary author of that recent paper in PRL that I just mentioned. So with that, thank you for your attention, and I'll be happy to have any questions that came up in the talk.
So can I start? Thank you so much for the talk. I was wondering whether you have some intuition of, I mean, you talk about plasticity, which I think it's already like a partial answer, but you have some kind of intuition of why these hybrid cells are better at initiating a new colony. Is like also like, I mean, could it be regarded as some kind of overcoming a nucleation barrier or something like this?
I don't. First of all, it could be. I mean, so there were sort of different ideas out there. So when people first started thinking about the fact that these clusters were more efficient at generating metastasis, there's sort of two ideas which are not mutually exclusive, but there are sort of two ideas. One idea is that, you know, it's good to have friends when you move to a new place. So if you move to a new environment and you bring along 10 other cells of your own type, then you can sort of shield each other and you could begin to grow, because in some sense you can surround, it's sort of like you've passed the nucleation barrier. So if you, one cell by itself is too surrounded by a foreign environment, but by the time you have 10, then cells mutually reinforce each other. That's effectively like a nucleation barrier. It's sort of like saying that a solid seed and a liquid, if you have enough of the solid cells, then the penalty you're paying for the extra surface energy is balanced by the positive interactions with the other cells of your type. So the nucleation barrier idea is one of the ideas that says that bringing along a number of your fellows to help you will enable you to grow more quickly. So I don't think that that's necessarily wrong, but I think there was also another aspect which was indicated in one of those biology papers that I mentioned very briefly, which is another possibility is that it's not about nucleation barrier, it's actually about the properties of the individual cell. And that was indicated in this paper by this 2019 PNAS paper by the Weinberg group, where they actually showed that it really, you needed to actually have cells with these specific properties of the hybrid cell. It wasn't enough to just have a large number of cells, and that the easiest way to get there was to create these clusters with hybrid cells. So they showed you really needed at the individual cell level the ability of the cells themselves to be adaptable. So that's not a nucleation barrier, that's a statement about a cell's capabilities. Now, if you say, what does it mean to be adaptable? So the only sort of knowledge we have about that really come from the stem cell field. In the stem cell field, you can ask, what is different about an embryonic stem cell versus a liver cell or a neuron or whatever? If you look at it, why are some of those cells very adaptable and can change in response to a whole bunch of environmental stimuli, and why are the other ones basically set in their ways? And the answer has a lot to do with what is the emerging field of epigenetic regulation. So epigenetic, in the context I'm using it, means that you set up a state of the system with these interacting transcriptional factors and expression, and then it gets locked into place by actual physical structures of the DNA. So their genomes and their modifications of the DNA, and it gets folded into active regions and inactive regions in the chromatin, and it gets marked in terms of genes that are able to be expressed and genes that are basically turned off semi-permanently. And so the idea is that stem-like cells, which is, as I said, the only analogy we have to go with, if you look at them, always have much more open chromatin. They always have many, many more. They have much more noise in their transcriptional profiles. So they're in a state which is much more fluctuating. It's in a state which is much less, it's sort of like being at high temperature in some type of potential well. So they have large fluctuations. They have lots of genes being expressed. They don't turn anything off. And so going into a differentiated state is like lowering the temperature and falling into one of those wells very deeply, and then you can't emerge. So I think that that's just as likely to be, at least a major part of the answer is that these hybrid cells have a much higher probability for creating this sort of cellular level plastic states, which have these large fluctuations, have these exploratory capabilities, and can therefore find, other types of ways of organizing their networks in order to survive these adverse conditions. So I think both ideas could be there, but the idea about plasticity is not really so much about the nucleation. It's really more about what individual cells can do in this context.
Thanks a lot.
Sure.
Herbie, there is a question in the chat. I'll read it out to you.
Maybe a person could just unmute themselves and just ask it.
The question is, could you please elaborate on the biological nature of frustration?
Yeah, so frustration in this biological context is you have, gene A left to its own devices, upregulates gene B. Okay, so you would expect that in a typical state of the system, if gene A is high, you'd expect gene B to be high also. But then there might be some other interaction because, of course, this is some network. This is not just a linear pathway. There's some other network where gene A is activating gene C, and gene C is also influencing gene B. So if I think about this as some type of loop, well, if gene A is activating, if A is activating B and A is activating C, then if C is inhibiting B, then that's a frustrated loop. And that I can't come up with a consistent picture of how A is affecting B downstream. Because if I look at one pathway to get from one to the other, it's an activation pathway. If I looked at a different pathway to get from one to the other, then its net effect is inhibitory. So the question is, how many of those frustrated loops exist in a real biological network compared to what you would expect if you took the same network with the same in and out degrees and all the things that people who study network topology would have told you are the critical elements of the network structure. You fix all those, and then once you fixed all those, you then randomize the network and you ask, how many of those different pathways would I expect to be frustrated versus not frustrated? And the answer is you do that simulation and you get some answer, and then you compare that to the actual network itself. And in the actual network itself, what we discovered is that there's many, many fewer of those frustrating loops. So the actual network, the different ways in which you can get from gene A to some downstream effector are relatively coherent with each other. Not always. In fact, we found specific cases where that wasn't the case, but just as an average structure of the network, there were many, many more reinforcing interactions than competing interactions. So in a system which has many, many more reinforcing interactions, what you expect is that the states of the system can be much more neatly, subdivided because, if A and B are always correlated by all pathways, then I expect to find, mostly states where A and B are either both up or A and B are both down. I don't expect to find that many states where A is up and B is down. So if I look at this type of frustration, I can understand how the network gives rise to a much smaller number of states than one would get by just assuming a random network of size 25 or 75 or something like that. So the impetus for this paper was a paper in PNAS by an Italian group, which looked at a 76 node network and basically discovered that in the 76 node network, if they just looked at all the possible states, they would find like hundreds of thousands of different states. And we thought this, it doesn't make much sense. What good are all those other states? So what we discovered was that, yes, there were hundreds of thousands of those states, but if one looked specifically at states that were sort of representative of the actual topology of the network, looked for low frustration states, these, first of all, would exist, where they would not exist for a random network. And not only would they exist, but if you would, for example, ask questions such as, let's say I start with random initial conditions and measure the basin of attraction of the different attractors of these states. Well, these low frustration states had essentially took up all the basin of attraction. So starting from almost any random configuration, you would find your way into the basin of attraction of one of these specialized low frustration states. So it basically meant that even though these states exist in some, general, in some mathematical sense, in terms of their ability to affect the dynamical progression of the system, they're essentially not relevant compared to these states that reflect in detail the specialized topology of the states. I don't know if you went to the talk that Mohit gave last week someday, Wednesday, I forgot what day you gave that. But that type of property, well, he also discovered in a different phenotypic transition, not EMT, but related to different types of cells that can be evolved in lung cancer. And so we don't understand that, whether this is a general property or just of some specific types of cell fate networks, but that's what we mean by frustration in these models. Okay, I hope that answered the question. What's the next? The hybrid maybe leader cells more elongated or polarized compared to epithelial cells, which might be round. Yes, I mean, they're sort of halfway. So if you look at them, so for example, I'll give you an example of differences between hybrid cells and epithelial cells. So if you look at collective motility and the cells are moving collectively, then, at first glance, the cells look similar because, okay, epithelial cells can move collectively. Certainly people who've studied vertex type models, whatever, and other experimental systems, I've seen cells that look very epithelial that move collectively. But if you look in more detail, what you discover is that, epithelial cells have all sorts of adhesive proteins besides cadherins that really make tight junctions with their neighbors. So for example, if you go and measure the flow of material through an epithelial layer, what you discover is that it has very low permeability because the cells really are at the nanometer scale. The membranes are just attached to each other via all these different proteins. But if you study hybrid cells, even though, it looks sort of not so different, they look a little more polarized. They certainly don't have up and down apical basal polarity, but they sort of look the same just on the microscope. But if you study, for example, the permeability going through a cell layer of hybrid cells, what you discover is the cells have cadherin bonds, so they're still sticking to each other dynamically, but there are large gaps. The membranes are delaminating from each other because they don't have these very strong tight junction proteins, and now there's a large permeability going through the layer. So there are direct morphological differences between cells that remain strictly epithelial and ones that become hybrid. Now, leader cells are a separate category of cells that I think we don't completely understand yet. Is one of the things we've been thinking about for this past year is, exactly what is the nature of leader cells, and it seems like there are multiple possibilities. One possibility is that leader cells can be cells that look hybrid in terms of their gene expression patterns. And those cells do look different. Those cells are bigger. They have stronger adhesions. But exactly how that's connected to these molecular features of hybrid EMT cells, I think is still not completely understood. So the answer is yes, they do have specific morphological differences that distinguish them from epithelial cells. And one can look at those things experimentally in addition to looking at the gene expression patterns. And they do play a role. These features do play a role in cell motility directly. You do see different motility patterns, but exactly how that enters into the specific case of leader cells, I think, is still a subject for research that we don't completely understand. I hope I answered that question. If there's any follow-up, please just unmute yourself and ask. Okay, another question. So where do we go? The other question I can paraphrase as, where do we go from here? Well, I mean, of course, the answer where we go from here is the multiple ways of proceeding. So I'll just explain a few ones that we're trying to understand. So in no particular order, one of the things that we've gotten very interested in based on the literature in cell motility and based on a general literature in the cancer biology world is, how does cell metabolism connect to these phenotypic transitions? So there was this idea in a paper, I don't know, by now 10 years ago, that cells somehow organize themselves in a modular way, where they have different subsystems and each subsystem does its own thing. And then there are sort of weak connections between those different subsystems. So even though this paper appeared in Nature, written by a bunch of really, really famous people, I actually think it's probably almost completely wrong. So when we look at things like the EMT circuit and we look at how it affects cell metabolism, one sees that there's a huge forest of different connections between the two. Now, cell metabolism for cancer cells usually is phrased in terms of how much of their energy are they getting from using the mitochondria versus how much of their energy are they getting from doing simple glycolysis, which means basically just taking sugars, glucose, and partially catabolizing them and then secreting the rest as lactate. And there's a long history in that problem, but the point is that when cells start to move and start to do different things, they have different metabolic needs. So cells in their epithelial state are devoted to growing as quickly as possible. And that places certain needs on the metabolic system. Essentially, you need to build new cell material, new biomass as quickly as possible. So that's one metabolic constraint. Cells that then start moving, and in particular when they start moving through complex materials, they need more energy because, and this has been shown, for example, for leader cells. Leader cells switch their metabolism to states that enable them to get access to more ATP because they need to secrete enzymes to basically blast their way through the medium. They need to actively move their cytoskeletons. Moving is a highly active process with lots of ATP needs. So one of the things we've been looking at is how do these different cell types reorganize the metabolic systems of the cells. And the reason for that partially is we keep looking for handles for what are unique features of these hybrid cells that would enable them to be targeted versus other cells. So if we could find some unique ways in which these hybrid cells differ in their metabolic needs compared to normal cells, compared to other, even the primary tumor cells, that would give us some leverage over what types of treatments might preferentially affect those. So that's one direction. Another direction is all of our cell motility models are trivial in the sense of they're all based on experiments in the biophysics community, in the active media community, of cells moving on surfaces. And of course, in real metastasis, cells don't move on surfaces, they move through fibrous networks. And so there's an interesting challenge of seeing how one can understand the interaction of the cell motility and what we saw in the previous talk, the cell-cell adhesions and how they stick to each other and coordinate each other's together with the cell matrix adhesions. They have to move through this fibrous material, which has interesting mechanical properties, which of course limits the size over which cells can squeeze through. And we want to be able to extend our ideas about cell motility to understand transitions between single cells and collective cells, also in the context of real materials that the cells go through. And that's another area which we're pushing. And of course, the third area, which I didn't mention at all, and if maybe the next time get invited to hopefully an in-person meeting in Bangalore, I will talk about that instead, is that in the modern cancer biology world, there's been a huge shift of emphasis to where what most people think is the most promising way to attack metastasis or any other aspect is to activate the immune system to attack the tumor. And so there's a whole new set of questions that arise when you ask not about how cells are able to start growing in a new environment, when cells are able to actively move through environments, when cells are able to resist previous generations of drugs. There's a whole new set of questions when you begin to think about how cells can resist being attacked and destroyed by the immune system that under a variety of circumstances can clearly figure out that cancer cells are foreign bodies in your foreign objects in your body. And one of the interesting aspects that we've been learning is that hybrid cells, again, because of their abilities to sort of react to environmental systems are able to also somehow suppress immune responses. And we don't understand really why. We don't understand why properties that I've described so far that we understand for these cells, or at least conceptually understand. We don't really understand why that also goes together with being able to evade immune system detection. But we clearly see evidence of that in experiments done by our collaborators, and that's, I would say, for the longer term, that's the most interesting question that we don't yet understand. So, we're sort of motivated by the things we understand the least, and from my point of view, that's the thing we understand the least.
But there's one more question in the chat earlier. Do we have complete information about the signaling molecules of EMT?
Yes. So the answer is we have information. I mean, complete is of course a word you should never use in biology, because they always discover new molecules and new examples. But we certainly have information about a large number of external molecules which drive this. The one that's used experimentally in many systems is called TGF-beta, which is some signaling molecule that's well established, and many cells, maybe not all, but at least a wide variety of cells, if you actually give them TGF-beta, will in a laboratory setting or even in a 3D setting will undergo the transition. So TGF-beta is certainly one of the most commonly used molecules that will give rise to this transition, and one of the ones that's believed to be important in different in vivo contexts. I showed you the example briefly of the Notch signaling system. The Notch delta system is a very interesting signaling system where it's sort of like the previous talk, where you have one protein molecule on one cell and it interacts with another protein molecule on the other cell, but these are not just responsible for adhesion. In that case, that interaction between those two molecules actually then causes a signal to be sent into the Notch-rich cell that actually then, of course, does the same thing as all the other signals, changes gene expression, alters the cell fate. So the Notch system is another example which you can use to initiate this epithelial-mesenchymal transition. There are also various growth factors that seem to play a role. So the answer is we have a large number of different signaling modalities which will give rise to this transition. The most convenient is TGF-beta. It's not the only one. And experimentally, laboratories that study this either use TGF-beta or one of the analogs, or the other possibility, of course, is you can go and just upregulate genetic by CRISPR or just some type of more benign chain in terms of upregulation of one of the stages of this progression, and then that will drive the transition. So you can upregulate, for example, snail via genetic means, by genetic engineering means, and then you can drive the rest of the transition. So I think there are a variety of methodologies which enable you to get access to this transition in an experimental setting, which people have used. But probably there are others that have yet to be characterized. But yeah, anyway. Well,
in case there are no more questions, thank you once again, Herbie, for an exciting talk, and I will